Our group analyzed the article Cell Metabolism, a predictive model for selective targeting of the Warburg effect through GAPDH inhibition with a natural product. To approach this study, researchers used the natural product koningic acid to see if it would inhibit GAPDH, which is said to play a role in the Warburg effect, and to determine if the status of individual genes determines koningic acid efficacy on the basis of targeting the Warburg effect. The results prove that Ka does indeed inhibit GAPDH, only in cancer cells exhibiting the Warburg effect, which can be done in vivo safely in human cells. However, it is important to keep in mind that not all cells display the Warburg effect. This can provide another tactic to attack cancer in a way that's not just targeting its genetic makeup.
I'm now going to talk about exactly how the experimenters carried out this study which is called a predictive model for the selective targeting of the Warburg effect through GAPDH inhibition with natural product. So let's first break down this title. What does it mean? So it's a predictive model, so they're not sure if it'll work, for the selective targeting of the Warburg effect. The Warburg effect is a metabolic pathway that is seen in cancer cells whereby they mostly only use glycolysis and lactic acid fermentation to produce ATP, which is not the most reliable way, but we'll get into that later, through GAPDH inhibition with natural product. That's the final part of the title. And what that is GAPDH inhibition. There's an enzyme called GAPDH in glycolysis that they found a way to inhibit it using a natural product, which is just an acid called koningic acid, or Ka, found in the T. koningi fungus. So they found a way to use a natural acid to inhibit an enzyme used in the metabolic pathways of cancerous cells that will thus limit the growth of cancer cells while also sending off a red flag for cancerous tumors. So let's look at figure 1a. And you can see in figure 1a the whole glycolysis and lactic acid fermentation reactions. And you can see the enzyme GAPDH about halfway down on the right. In blue, it's an enzyme along with metabolites that are used in this whole metabolic pathway in cancerous cells. And then if you look at E in figure 1, you can also see that compared to cancerous cells and non-cancerous cells, only cancerous cells are affected when you limit their GAPDH. So normal non-cancerous cells aren't affected by koningic acid. They carry on with their daily lives. They aren't really affected. They don't care much, which is why it's called selective targeting in the title. And this also shows that this whole GAPDH inhibition using koningic acid can be achieved in vivo, and it has been achieved. The experimenters injected both the acid and the plasmid from the fungus into both mice and a human cell, and they both survived. It didn't affect them as long as they weren't cancerous, so it is safe to use on living humans.
So how does this connect to biology? Ecology is broadly defined as the study of how organisms interact with their environment. The proximate cause of a behavior analyzes the how behind an action, while the ultimate cause of a behavior analyzes the evolutionary why behind a behavior. In this case, T. koningi fungi secretes Ka to ward off competition like anything around it that eats its sugar source, which is the proximate cause. The ultimate cause is to be able to live and reproduce. Biological evolution is basically defined as the change in heritable traits of a population over succeeding generations. This particular research study revolves around the natural selection mechanism of evolution, which is the idea that certain genotypes produce more offspring or have a higher fitness than other genotypes. By chance mutations, the fungi that evolved this ability to secrete koningic acid was able to survive and reproduce by warding off anything that threatened its food source. This article also mainly concentrated on glycolysis and cell metabolism. Normal cells perform oxidative phosphorylation in the mitochondria to produce energy, and through this process produce 36 ATP. However, cancerous cells perform glycolysis, which occurs in the cytoplasm and only produces 2 ATP, as well as lactic acid fermentation, where pyruvate is reduced to NADH. Both of these processes are anaerobic, but cancerous cells are not seen performing oxidative phosphorylation, even in the presence of oxygen. By accelerating glycolysis, cancer cells outperform normal cells to get sugar. So why is this study important? Researchers found a way to non-genetically target cancer using a natural product that can be achieved in vivo, which means it takes place in a living organism. This is a huge step to cancer research in oncology.